Okay, plan for this afternoon then is to sort of pick up where we left off. I'm going to do a very quick recap of what we did last time, and it was our sort of introduction to the building materials of the nervous system. And then we're going to look at, we've done the action potential and, no, resting potential and the action potential, haven't we? What we need to do today is synapses, which is the connections between nerve cells. And we may get a chance to do a bit on the brain and just look at different areas of the brain and what they're responsible for. And then tomorrow morning we will go on to a little bit about the spinal cord and reflexes, a bit about nerves, well, one aspect of nerves, the autonomic nervous system, that's the sort of homeostatic bit, and then hopefully make a start on the eye. That means we will end up, because that'll be our last bit of biology, we'll end up leaving the eye in sort of mid-air, as it were. But we'll have to pick that up again after Christmas break. And then tomorrow afternoon is the opportunity to really get that assignment, the chemistry assignment, as done as possible, because that's due in on Friday, so that you can just chuck it in the box and go off, and that's that. All right? So you certainly can ask.
We haven't met criteria for that.
Yeah.
Will we be obviously that to be
Yeah, if you haven't met the criteria on that, it's the same as a graded assignment in that you get a resub.
Yeah.
And so, yeah, I mean, I'll turn them around over Christmas, or, you know, of 5 minutes here or there. And, yeah, so anything that needs to be finished off. So, you know, even if you feel you can't get it completely finished tomorrow, if there's a section you just can't face, you know, if the calculations have actually sort of, you know, mind you, I think a useful thing tomorrow is, you know, I will go over this, we'll go over the calculations again. And so, yeah, I'd hand in, you know, what you can. And even if you know you haven't passed everything, that's fine, because, yeah, you get another go. So, yeah, and there's no way that, you know, we let people fail on ungraded assignments. So, right, so our introduction, let's just whiz back a bit, see where we've been with the nervous system. We had an overview, and I'm going to keep coming back to this slide because we're not looking at this yet. We're just looking at these neurons, the excitable cells that all this lot's built out of. And what we've got to do is to sort of set that up so that we can see how these things work. And what I'm aiming to do, therefore, is to come back to the brain over there on the right as an illustrative bit of the central nervous system. I'm doing it sort of backwards across the board. And then tomorrow morning, we might touch on the spinal cord a bit. We'll see how time goes. Then we'll look at the autonomic nerves under the nerves section there. And then out of the sensory receptors, we'll deal with the eye. And as I say, that could be left in mid-air. And so hopefully then, you know, we'll have a sensible place to leave it for Christmas anyway. From there, we went into the different sorts of neurone. So we've got the sensory, the motor, and the relay ones. We had a look at the technical bit, the nerve impulse, which consists of the resting potential when the cell's not conducting anything, and then the action potential, which is the basis of the nerve impulse. And yeah, that's rather beautiful, but you've got that in your handouts. We looked at how the different factors can affect the speed of a nerve impulse, and then you did the practical to see how fast your nerves were working, and all turned out to be pretty normal, surprisingly. The next thing then is to look at how do we get a nerve impulse to go from one neuron to the next, because if you are connecting up parts of the nervous system, so let's say you're connecting the eye or the receptors in the eye to the optic nerve to the visual area of the brain, the visual area of the brain to some other area that carries out perception of what you've seen. How do those all connect? Well, given that most people think of nerve impulses being like electricity, you would think, well, you've just got to bring the nerve cells together so they touch and then it can be passed on, which is wrong. All right, so there is no contact between nerve cells. Now this makes it all sound a bit strange, doesn't it? Because how are our nerves communicating if none of them touch each other? And how do they manage not to touch each other then? A lot of the not touching is because of the myelin that's wrapped around them. And certain parts will touch, but there is like this packing tissue called neuroglia that just sort of holds them in position. But whereas with an electrical cable, if you put electrons into one end, then those electrons will push electrons through the metal that makes up the wire and push electrons out of the other end. That's electricity. So, you know, you then connect that up, the electrons can move into something else. With nerve cells it is not the same, but we have these special little structures called synapses. And synapses are reliant on the movement of chemicals from one cell to the next. And what we're looking to do is to get the action potential, the depolarized membrane of one cell, communicated to the next cell. Now, these are not continuous cells, so, you know, contact isn't going to help. You know, you've got a depolarized membrane, that's not going to depolarize the membrane of the next cell. So these are quite elaborate structures, but they are really interesting. And one of the most interesting things is the number of different chemicals that we are familiar with or aware of that actually interfere with the way that synapses work. So we'll come back to that in a bit. You've got a good diagram of this in the handouts. All right, synapse structure and function or something. So I'm going to build my own version of that, label it and explain what happens in stages. And then we'll have a look at the factors, well, sorry, the substances that can influence synaptic transmission. All right, so push that out. It should look like that. All right. I mean, you can colour code it like the other one if you feel that way inclined. Sorry? Ah. Right, synapses then. Yep, neuroscience is the study of, yeah, neuroscience is really interesting. It's nerve cells, it's the large scale stuff in the nervous system, it's the way substances influence the nervous system. I've had people go off and do degrees in neuroscience off this course, but they are not locally, this is the thing. I think I had one go to Bath or Bristol or somewhere like that, another one go to York, and they didn't require especially high grades, interestingly. You know, you would think they would, but no. It was, yeah, they're different offers. Yes. You'll be leaving at 25 past, not in 5 minutes. Okay, all right. Well, it was nice to see you. I just need to nag you about your UCAS. What state's it at?
I've just got to put my personal statement on because
Yeah. Yeah. Okay, all right. Right, a few more nags to do, I'm sure. Right, a synapse. Well, the diagram's going to develop and it's going to fill this whole panel. We start with an incoming nerve cell, incoming neuron. There's its axon. And then the end of this neuron has an enlarged bit that has a variety of different names. I tend to these days just revert to a sort of simple option and call it an axon terminal. All right, so this is my axon terminal. You will, however, see it, I think, labelled in that diagram in your handouts. Is it the synaptic button or something like that? No, synaptic cleft is going to be the gap between. I don't think it probably isn't labelled at all then. The normal name for this, or the name that we've always used in the past, is synaptic knob. Yeah, well, vesicle. The vesicles are the neurotransmitter, but the actual enlarged bit, I've made it very lopsided there, that enlarged bit there used to always be called the synaptic knob, which was a very good excuse to stand at the front of a class and say knob several times. So, yeah, I mean, this is closely followed by cleft, of course, you know, the synaptic cleft. But the synaptic knob, it just means this enlarged bit. But axon terminal just sort of, you know, gets you out of that. The axon coming in, of course, will probably have myelin around the outside, so we put a bit of that in, which stops as it gets to the end. But what this means, of course, is that the action potential is going to make its way. Let's colour this. The action potential has come from there to there, then from there to there, then from there to there. All right, so the action potential, I have not written, I've left enough space for that. So the action potential is jumping down the length of the axon, and it arrives at the terminal. And it will have an effect there that will enable the terminal to pass on the message. Oh, I can't believe I called it a message. We need a few other bits here before I carry on with this. Now, you noticed inside that you've got these little round things. These are vesicles. A vesicle is a bit of membrane that encloses something. So these are vesicles of neurotransmitter. Now, a neurotransmitter is a chemical. It's today's big word. Neurotransmitters are chemicals which enable the impulse to be passed from one cell to the next. So they do what they say, really. And there are about 50 different ones in the nervous system doing different jobs. There are some that you might be familiar with. Have you ever heard of dopamine? Yep, that's a neurotransmitter. Have you heard of serotonin? It's a neurotransmitter. You're less likely perhaps to have heard of some of the others. Acetylcholine is the most common one. You'll have heard of adrenaline as a hormone, but noradrenaline is a neurotransmitter that works, not surprisingly, like adrenaline. And you may have come across one called GABA, gamma aminobutyric acid. You see them on the sheet. You don't have to know all of these. I'll give you a selection. But this neurotransmitter, like I say, is about 50 different ones because they do different jobs in the nervous system. So some of them will stimulate some nerve cells, some will inhibit nerve cells, you'll have 2 or 3 options at one synapse sometimes. And so I'm going to put some little dots in there. Okay, so this is our neurotransmitter. And then you might realise what's going to happen to it. It's actually going to leave that cell. But I'll come on to that in a minute. There's something else in the axon terminal that we are very, very familiar with, a word you've tried to blank out of your consciousness. That's the one, it's mitochondria. Because this is quite an energy-intensive process. The bit that involves most energy is actually trying to take the neurotransmitter back in and recycle it. So we've got a mitochondria in there. Now, this is our incoming neuron. We tend not to use that word. The word that's more appropriate is afferent. Afferent means coming in. And it's a word we meet again in the kidney, which has blood vessels coming in and going out. We have afferent capillaries and efferent capillaries. That's our afferent neuron. You could call it the presynaptic neuron because it's before the synapse. This whole structure is the synapse, and that neuron comes before it. That's most useful here if you're just talking about the membrane. So if we refer to this as the presynaptic membrane, that's fairly self-explanatory then. Okay, so that's what we've got coming in. Now, what about the going out then? This is harder for me to draw, but you have got the diagram in the handouts, which, yeah, you can see on the next cell, a bit underneath the incoming, you've got some little square-shaped things with a sodium ion shown passing through one. So those are sodium ion channels. Now, we've already met these, haven't we? In the action potential, resting potential and action potential. It's facilitated diffusion, and you have the sodium channels and the potassium channels. So this next membrane is going to have sodium channels in. Now, that's fair enough because you want to depolarize that cell, so you need sodium to rush in. But what's going to open those sodium channels, do you think, perhaps? Something's got to act as a stimulus and open the sodium channels. The neurotransmitter, yeah, that's what happens. So we have to have receptors for those. So this ends up drawn way out of proportion, really, that I have to have like a little cup shape for the receptor, and then that's coupled to a sodium channel. So a little cup and then a channel. A little cup and a channel. Whoops, made ridiculously long channels here. All right, but that's the general sort of idea there. Let's trim those down a little bit. I got a bit carried away there. Right, so the little cups I've drawn are receptors for the neurotransmitter, and the little channels are sodium channels that are going to be opened when that neurotransmitter binds to the receptors. Okay, so these go in there and in there. All right, so let's, whoops, that's where they go. I won't put one at the end because I don't feel like it. This is our postsynaptic membrane. And the other things I could label on here, this gap between the two cells, absolutely microscopic. You know, we are talking about, I don't know, single numbers of micrometers probably. I'm not sure. One of those things I ought to know but I don't. But this gap between the cells is critical, and it's called the synaptic cleft. I'm not going to label the receptors because it is fairly obvious there, and I'll write about them in a minute. But we've then got those receptors are coupled to the sodium channels, so that these things here, that and the other ones, all right, so that's a sodium channel, so that when the neurotransmitter crosses the synaptic cleft, binds to the receptor, it opens the channel, sodium rushes in, depolarizes that membrane. Now, I have missed out the beginning bit. I haven't said how that happens, you know, because what is it about an action potential that makes this happen? Well, when the action potential arrives at the axon terminal, that also opens some channels, but these are ones we haven't met yet. They are calcium channels. So one of the reasons that we need calcium in our bodies, apart from the obvious, which is, that's it, bones and teeth, we also need it for blood clotting, but we also need it for synapses to work. So when the action potential arrives at the end of the axon, that causes these channels to open and allow calcium ions to go in. When the calcium ions get in, that makes the vesicles move to the presynaptic membrane. They then release their contents by the process of, having just done cell transport processes, what does that? Exocytosis, yeah? All right, it's being released out, exocytosis. It's a vesicle. You wrote about vesicle transport. The transmitter then diffuses across the gap, binds to the receptors, opens the sodium channels. The sodium channels let sodium in, that depolarizes the membrane. Bob's your uncle, it's been passed to the second cell. Now, what's to stop that just firing off again and again and again and again and again? You actually have to remove the neurotransmitter from the cleft. Otherwise, it will just keep binding to receptors and firing off an action potential, and again and again and again. So this is all happening in milliseconds. All right, you think about the fact you've got that diagram, that the graph of the action potential was 2 or 3 milliseconds. This is going to be even less than that, where you've got the neurotransmitter released, crossing the cleft, and then being removed. It has to be removed and probably recycled. You know, why would you waste it? We have enzymes that move in and break down a lot of neurotransmitters, but we also have a way of taking them back up again to be reused. Right? You're not going to waste stuff that you've made if you can reuse it. So that has to be done really quickly. And you can interfere with that process. And we will look at some of the ways of interfering with this whole thing in a bit. So what I'm going to do now is to go through it step by step in a sort of numbered list so that you get the sequencing right on this. And then we'll have a look at sort of where we could interfere with it. So I'm going to try and remember to put everything in this. And starting with number 1 would be the action potential arriving at the axon terminal. It's this wave of depolarization. The depolarization opens the calcium channels and calcium moves in. So action potential arrives at axon terminal. And then calcium ions, I'm just writing like that, Ca2+. Calcium ion channels open. And calcium ions move in. Okay, so what this is doing, it's changing the electrochemical balance in the cell. All right, so and this is why you do chemistry. So it's to understand how important some of these things are. Sodium ions, what are they? Where are they? You know, what do they do? Potassium ions, similarly. Calcium ions, similarly. You know, it really sort of makes a difference if you know what an ion is. Or even if you've got a vague idea, it'll do. So the action potential arrives. Calcium channels open, calcium ions move in. The vesicles of neurotransmitter then move to the presynaptic membrane and, what shall I say there? Release their contents by exocytosis, probably. After using the word once, I think I'm going to go for an abbreviation of neurotransmitter because it is a pain in the neck. So, vesicles of neurotransmitter. I think my abbreviation will just be N/T. Move to presynaptic membrane. So this actually helps to keep the impulse going one way. So they move to the presynaptic membrane and release contents via exocytosis. All right, so you can see now the reason for doing cellular transport processes before we do this. In the resting potential, we had active transport and facilitated diffusion. Here we're going to have exocytosis and simple diffusion. I'm sure somewhere in all this there's active transport as well. Oh, there'll be probably endocytosis as well to take it back up again. Not sure. That might be active transport. So we've now released the neurotransmitter, and so that, which I'm now just referring to as NT, neurotransmitter, diffuses across the cleft, all right, that gap between the two cells, and binds to receptors. Which are coupled to sodium channels. Okay, so far we've not managed to transmit anything, but the sodium channel's now open. Sodium ions rush in. Whoops, I don't need to write ions. And depolarize the postsynaptic cell membrane. In other words, what we've got here is action potential transmitted at this point. So that's what we were aiming to do, to get the action potential from one cell transmitted to the next. So it's happened, but we've now got to stop it. And so we need some way of removing the neurotransmitter from the cleft. So neurotransmitter removed from cleft, possibly by enzymes, we'll assume so. That might be breaking them down, but enzymes and transporters, or transport proteins, probably, which take it back into the presynaptic cell. Okay, so you can then send another impulse down and get that passed on. Now, I can't remember whether it's still in the exam, but I always used to ask a question about explain how it is a synapse maintains the one-way direction of a nerve impulse, and this is it. You know, that it's only the presynaptic cell that releases the neurotransmitter. It's only the postsynaptic cell that has receptors for it. And I think it may be one that I actually use in the merit distinction section in a particular example that you know, you can apply that to, but you know, you can only do this one way round. The postsynaptic cell doesn't produce neurotransmitters, so it can't send it back again, and the presynaptic cell doesn't have any receptors, so similarly, you know, it can't receive. So the impulse is only going from the presynaptic cell to the postsynaptic cell. Now, it may be you could have a situation where, if this is your outgoing cell, well, let's draw it a bit bigger this time. You could have, you know, if this is the cell body of an outgoing cell, you could have different nerves coming in with different neurotransmitters. So there's one coming in that makes red stuff. Here's another one coming in that makes green stuff. And these might all, there's another one. All right, so these are separate incoming neurons. You could have 3 different neurons connecting with one cell. I mean, there I've drawn a total of 7 synapses. 2 red ones, 3 green ones, 2 blue ones. I mean, they'd be covered in them. But it might be that the red incoming cell says, stop, don't send this nerve signal any further. It might be that the green one says, go, you know, pass this on, and it actually depolarizes the cell. And it might be that the blue one says, we need to wait for something else to happen, and then when that happens, it gets passed on. So it provides a degree of control in the nervous system, and this is where you would potentially use different neurotransmitters. So you might have, I'll give you a list of those examples, so e.g. neurotransmitters, and out of about 50, I'm only going to give you these 5. The most common one that's in most of the nervous system is called acetylcholine. If I could spell. You have noradrenaline, which is actually pretty much the same as adrenaline. And there is a bit of an overlap between the nervous system and hormones in that. It's just that they work in a particular part of the nervous system. Sorry, that does. The other ones that you've probably heard of, you've got dopamine. I think you've been talking about the reward pathway in the brain in psychology. Yeah, it's an interesting thing. Serotonin. I mean, dopamine's an interesting one. When people know about its role in the reward pathway, you know, it's sometimes the happy hormone, that sort of thing, because it gives you a reward for activities that basically keep you alive. So it triggers that pathway in response to things like eating, drinking, sex, and a few other things as well. Nurturing sort of processes. You get a dopamine reward for it. But of course there are drugs which can also trigger a dopamine reward, and that's part of the addiction process. So yeah, if it feels good, do it again, and it feels good, so do it again. And that's, you know, it's hijacking that reward system that's just supposed to keep us alive. But dopamine has another role in the brain. I'm not sure if you're aware of the other role that it has. A different part of the brain produces dopamine, which is responsible for controlling voluntary movement. I don't know about ADHD, but voluntary movement, you've got an area, there's a group of cells that produce dopamine that die off, and it causes problems in elderly people usually in terms of movement. Parkinson's. Parkinson's is the loss of a particular group of cells that produce dopamine. That dopamine is responsible for initiating movement. So people with Parkinson's have real difficulty. They freeze. And it's that initiating, apart from the tremor that we're all familiar with, you know, it's this inability to start a movement is the problem in Parkinson's. So consequently, most people who are on, who have dopamine, no, start that again. Most people who have Parkinson's are on dopamine. They take supplementary dopamine tablets, usually levodopa. And yeah, that apart from the other option is to actually stimulate those nerve cells with electrical wires put into the brain, which is turning out to be incredibly effective in a lot of cases. But it stimulates those because the dopamine's not being produced. Well, electric shock therapy really is like a turn it off and on again, to be honest, and that actually destroys some pathways. But the dopamine, the replacement of dopamine in this particular group of cells with sort of electrical stimulation, it just takes advantage of that similarity between nerve impulses and electricity. But it's a bit, actually, the most similar thing is what I was doing with my TENS machine. It's sort of the similar sort of thing that, you know, you interfere with the way that nerves work, or you replace the way that nerves work. ADHD, I'm not sure. Well, tics are usually Tourette's. So the thing is, a lot of those conditions are generally sort of malfunctions in brain chemistry of some sort. So, you know, it's a bit hard sometimes to separate them out. But sometimes they find strange things happen, don't they? You know, that there are unexpected effects, like giving Ritalin for ADHD, where it's actually an amphetamine, effectively. I'm not sure if marijuana helps with Parkinson's. Sorry? Yeah? Yeah? Yeah. Yeah, I mean, brain chemistry is a weird thing, and you've got to bear in mind that in terms of anatomy and physiology and stuff, I mean, if you took the heart, for example, they always say, you know, we know about 90% of what there is to know about the heart. There's only really 10% we don't know, and I should think it's even less than that now, to be honest, when you consider they can complete operations like the one they did yesterday. Was it yesterday? Oh, no, reporting on the little girl with the heart outside her body. But with the brain, it's the other way round. You know, they only know 10%, and there's 90% they don't know. So, you know, neurological processes are phenomenally complicated. And, you know, just the fact that we've got about 50 odd neurotransmitters, you know, it tells you how complex it is. The other one that I said out of this, I'll just give you its initials, and it's known as GABA. And people will usually say, oh, is that the same GABA as you find in... Sir? People usually say gabapentin. Yeah? Which is used for... I think it's used for epilepsy, but it's also used for really severe pain. Now, you can see why. GABA is a neurotransmitter. Gabapentin, therefore, has got something to do with interfering with a neurotransmitter. So in epilepsy, obviously, it's nerves firing off when they shouldn't do. And in severe pain, it's nerves firing off when you don't want them to. Yeah, all right, they should do. And, you know, there's issues with pain management that we actually are stopping our awareness of something that's trying to tell us there's something wrong. We don't deal with the thing that's wrong, we just deal with the pain that we get from it. So, you know, painkillers are a bit of an interesting thing, aren't they? So that's what a synapse is, and you will need to know that sequence. All right, so when we revise, we will go through this, and you can find a way of remembering it if you pick out key words in there. It is really just a matter of memorising the order of it. Now, if you have a look on the other side of the handout, if you look on, you've got synapse structure and function. On the other side, you've got some interesting stuff, which is the sort of applications of this. Health applications are always good. And now that we know how a synapse works, hopefully you can see there's actually quite a lot of places in that structure and process where you could stop it working or make it work more. You know, if we go back to our diagram, if you start, you know, where the action potential comes in, well, you could make the cell produce less neurotransmitter. You could make it less able to release the neurotransmitter. You could put a chemical in that's got the same shape as the neurotransmitter and therefore trigger a response in the next cell. You could block the receptors in the next cell. You could change the amount of neurotransmitter that was removed. So many things you can do with a synapse, which is why a lot of drugs target synapses. But they do very different jobs. So we're going to have a look at a few of these. I'm only going to sort of select highlights here. And I'm just going to take some of the boxes. And I'm going to start over the right-hand side, the second box down. Reduction in release of neurotransmitter. So you could reduce the amount of these green dots that was being released. All right, so it's either reducing the production of it or just the release of it. The first example in there is something called botulinus toxin. It comes from a bacteria called botulinum. I can't remember its other name. Oh no, it's Clostridium botulinum. That's what it is. It causes a severe food poisoning, severe to the point of death, because it actually paralyzes people and it prevents the release of acetylcholine. Okay, so it just interferes with transmission, so people become paralyzed, unable to move. That includes breathing muscles. That's your lot. However, how do we know that? How do we know of that substance, botulinus toxin? How do we know it better? Look at the first few letters of the two words. Botox. That's what Botox is. Yeah, Botox is botulinus toxin. That's what the abbreviation means. Yeah, people have that injected. People have that injected into nerves. So yeah, I wouldn't, I must say. Not, knowing what it can do. I mean, it's incredibly localized when it's injected, and it does wear off, doesn't it? Because it's not affecting breathing muscles or anything. It's just perhaps in wrinkly areas. But you end up with that rather sort of strange, smooth sort of effect, in people who should have wrinkles and should look like they've lived a bit, and don't. They look like some of those rather bizarre robots that they're talking about at the moment. So yeah, Botox is botulinum toxin. It stops the release of acetylcholine, so it stops muscles contracting and it stops wrinkles effectively. So yeah, I mean, how important is that in the world of medicine? Well, not very, unless you're a cosmetic surgeon down Harley Street or something and can charge hundreds of pounds for it, I suppose.
So if they got that Botox, they messed up.
I'm assuming that it's possible to mess up. I'm assuming, people have been damaged from it. Yeah. It is tiny amounts and it is very, very localized. This is the thing. But there are uses for it. Now the thing is, it's all very well saying, oh, I wouldn't do that. But it can be a matter of improving people's self-image, which can make a difference to their self-confidence, which can then make a difference to their entire lives. Well, could do. Yes. Now there are two examples, medical examples I was going to give you, and that's one of them. You sometimes get people with a condition called hyperhidrosis, which is overproduction of sweat from the glands in the armpit. And basically they're just, and hands particularly, actually, you're right, yes. Hands rather than armpits. Because the ones in the armpits are not actually the same sort of sweat gland. The ones in the hands, they are controlled by nerves. The ones in the armpits are more controlled by hormones, different sort of sweat. But if people have constantly wet hands, there's a real limit to the things that they can do in life. So sometimes you have the really extreme treatment for this, which is cutting the nerves that supply the sweat glands in the hands. I'm not sure whether it affects any other function, but to stop the production of sweat, they'll do that. However, they've now found that by injecting, and I'm not sure where, and it might be the injection I just remember somebody having injections in their armpit. That you can stop the nerves actually sending the impulses that cause the sweat production, and it's very effective. And I can remember one of those embarrassing bodies programs. I never really get those, to be honest. If you're that embarrassed, would you really go on telly? It's free treatment program, isn't it? But I can remember one of those, and I think it was somebody from locally around here, because one of my past students knew the person who'd had it done, who claimed she wanted to be air cabin crew, and therefore, excess sweat really wasn't going to work. And I think that was related to armpits as well, because if you put your arms up to the, close the baggage lockers and things, it was just, yeah. But it worked, it did the job, but it would have to be repeated. It does wear off when it's localized like that. The other application that I've never looked into in great depth, and whether it was only ever, I don't think it was just trials because I think I had a student whose son was offered it, and it's for children with cerebral palsy. Now, in cerebral palsy, you might be aware, walking is difficult because you get a contraction up the back of the leg that causes them to walk on their toes and toes turned in. You know, if you think about it, it's very distinctive, isn't it? That, you know, and you then don't get the proper gait as you walk. You know, you can't put your feet down heel first, and you can't put them parallel because they're going like this all the time. But that's due to contraction of the muscles up the back of the leg. Now, if you catch them early enough or at the right time, I believe, in some cases, if you use Botox on the nerves that cause that, the tension, you can actually get children to sort of put their heels down and develop some degree of normal walking. Because it's only temporary and it only really works early on, it's not a permanent fix, but it improves things like their posture and movement generally. So, you know, they do. Ah, right, yeah, yeah. Yeah, I think no, I think it's one of those things that you think, wow, that's a really good thing to do. Oh, right, quite possibly, yeah. People have got overproduction of saliva. Ah, right. Yeah, yeah. I think I have heard now of several more applications for it. So, you know, Botox is it's got its place in medicine anyway. So there's an example. Now there are some others in there as well, but we'll skim over those, or we'll be in this section forever. Although the last one in there, you can see the dark printed ones. Alcohol may cause a change in sleeping patterns, particularly increasing frequency of waking periods by reducing the synthesis and release of serotonin. So, you know, that's significant use of alcohol can do that. Not with everybody. I mean, this is always the way, isn't it? Next one round, decreased sensitivity of postsynaptic membrane. So you've got things like depressants that do that, barbiturates, which are sort of sleeping tablets really, aren't they, I suppose, best description, which prevent or reduce the permeability, the ion permeability of that membrane. So it's, you know, stopping those sodium ions being able to rush through. Down at the bottom right, altered availability of postsynaptic receptors. First example there is not generally a problem around Lowestoft. Curare, a poison used by South American Indians to tip poison arrows. So yeah, it's not something that you don't get that, you don't really get poison arrows shooting at you out in the bushes around here. Yep, yep. Yeah, rubbing them up. That's it. Rubbing the arrows on the frogs. Yeah. Curare, I think, I'm not sure. That's not a frog one. I think that's a plant-based one. And then there's other examples in there. And if you look further down, very high doses of nicotine actually do that. And very high doses of nicotine are fatal. I remember being at university, and a friend and I were doing a comparison of the amount of nicotine in several brands of cigarette compared with actual nicotinia plants, and we had to sort of extract stuff from all of them, and then we needed a baseline to compare against. So we had to have a solution of nicotine that we could get some results from, and then compare all our samples with that. And the solution of nicotine, we were third year degree students, we weren't allowed to make it up. It came in a bottle about a couple of centimetres high, and the lab technician had to make it up for us because one drop of that on your skin would kill you. So, pure nicotine is completely, lethal. So yeah, that's blocking acetylcholine receptors there. You come round, not the one at the bottom, but the one above that, inhibition of adenyl cyclase. I think I called it adenylate cyclase when we were doing the peptide hormones. But it was the second messenger thing. Now, if you inhibit that, if you inhibit the second messenger, then, if you inhibit, it's the breakdown of the second messenger, actually. This is a different enzyme, that's why I'm getting it confused. Adenyl cyclase breaks down cyclic AMP So it leaves it there and makes it has a stimulant effect, therefore. So you'll see examples in there. You've got caffeine. All right, caffeine, and I think the thiophyllene, that might be one of the ones in tea. I know you've got bromophylline in there, but I'm not entirely sure. But yeah, so, you know, tea and coffee are stimulants because they do that. If you come round to the left-hand side, mimicry of neurotransmitter. Well, nicotine is there as a mimic of acetylcholine. So it's a stimulant for, you know, low levels, puts your heart rate up, that sort of thing. And then there's a really confusing bit further down in that I think is written back to front. It says something about endorphins. Heard of endorphins? Yeah? Endorphins are the body's natural painkillers that we produce under severe stress, usually. And they are related to our need to carry on when injured. And, you know, you hear stories, don't you, of, say, rugby players carrying on and then finding they've got a broken bone somewhere, that sort of thing. And it would go back, I'm not going to use football players in the same scenario, you understand, because they will not carry on, they'll roll on the floor and, yeah. Yeah, definitely rugby players. It's also associated with people who get hooked on exercise. You know, who feel they've got to go for a run every day or they've got to go to the gym every day. I personally can't actually imagine what that's like, but you know, they get hooked on their own endorphins, all right, because you push yourself to this limit and then your body's trying to protect you, actually. And the origin of this would be, let's say you're being chased by a wild animal or some hostile person, you trip over, twist your ankle, you can't roll on the floor like a footballer, can you? You've got to get up and carry on. So your endorphins kick in, they let you keep going, and then when you're safe, you sit down and deal with it. So that's what these are. Now, the way it's written here is, it is back to front, because it says endorphins, such as, they mimic the opiate drugs. No, they don't. The opiate drugs mimic the endorphins, don't they? Right, the natural bit. Sorry? Yeah, happy. I've no idea whether they've got anything to do with bananas. Chocolate, definitely. Chocolate has a similar sort of effect. Now, what we're looking at, opiate drugs, we're looking at heroin and morphine. We're looking at exercise. We're looking potentially at some aspect of chocolate. We've got three options there for achieving the same effect, more or less. But on slightly different scales, you have to admit, you know, these are not quite the same, and you know, it does depend on personality to some degree, doesn't it, as to what your choice is. But all right, you've got, these are your options: heroin, serious exercise, chocolate. Yeah, you can see I'm the one that goes for the exercise, can't you? But but you know, the actual it's the mechanism more than anything else that's similar. They are not the same thing, but they work by similar mechanisms. So mimicking neurotransmitters, you know, and it's sometimes, you know, replacing one thing with another can work because they have a similar sort of effect in the body. Although I don't know that you'd manage to get heroin addicts to sort of go for a good long run, you know, to try and get the same buzz. But well, it would be good, yeah. Yeah, and yeah, I mean, exactly. It's the personality thing is your issue, and because usually by being users, they're not healthy. They're not going to be able to run anyway. So you know, they're going to have spent all their money on heroin rather than on good quality food. They're going to be spending, you know, then they are not the people who will go for a run. So yeah, you know, ideal world, you know, we've got all these treatments and plenty of chocolate. Well, that, you know, I don't know whether you could eat enough chocolate to deal with that, but. Yeah. Ah, right. Yeah. I mean, if people are prone to addiction, then they could easily, yep. Or chocolate's the other problem, I suppose. You know, because you know it does have. I mean, there is this issue, and I know you discussed it with Sarah about, you know, what's the difference between physical addiction and addictive personalities and all this sort of stuff. And there is no one answer, is there? You know, there's an awful lot at play there. But yeah, you know, it's it depends on your definitions and all sorts. So that's, yeah, an interesting one then, mimicking neurotransmitters. And then you have, up at the top left, or second at the middle on the left, limited removal of neurotransmitter. Going through there, they've got cocaine in there. Acts as a euphoric drug by inhibiting the reuptake of noradrenaline. I would say more than that, it inhibits the reuptake of dopamine. Really significant in the reward pathway, that it inhibits the reuptake of dopamine, leaves the dopamine in the synapses, and so they keep firing off. So you just keep getting the reward from it. So it gives you a high. This is good. Keep doing it. Keep doing it. And that's sort of, you know, you can find good images of that. I actually got some really good ones. It was part of what I did on the masters was about that. But it's yeah, taking the dopamine. It depends on your definition. I was talking to Sarah about it. It's not a physical, but it's neurological. Now, to me, that is physical. So it's, you know, no, it's neurological is yeah, is sort of considered separately. But when you're a biologist, that's, you know, because neuro stuff is part of your physical or your physiology, it's actually quite difficult to separate that out. But yeah, it's not got quite the same sort of effects. So
It's not physical addiction.
yeah, but it is neurologically, because it's actually triggering the dopamine reward pathway by leaving the dopamine in the synapses. So yeah, you're still getting that reward for doing it. So, you know, you can be addicted through that mechanism, can't you? So, but definitions, you know, physical addiction, neurological addiction, is just, yeah, as long as you understand what you're talking about, I think is the important thing, isn't it? Because I mean, the word is much abused, isn't it? Addiction, you know, people use it for all sorts of things, and it's just important to be clear what you mean, I think, really. We've got any others in there? Some of the weed killers and insecticides inhibit acetylcholinesterase, which is an enzyme that breaks down acetylcholine. So that leaves acetylcholine in the synapses, so they can be toxic that way. That's probably how they work. Amphetamines come in that category as well. So, you know, it's really quite interesting. I think this is, you know, one of the most interesting bits about the nervous system, is looking at how you can interfere with synaptic transmission. So, you know, most of you will come across, well, I mean, even giving somebody painkillers is interfering with synaptic transmission. And, you know, you can usually find some, yeah, aspect of medication that most people have used that works at synapses somewhere. Okay, now, that's the bit that sort of finishes off the basics, the looking at the function of the excitable cells. And our next job is to start looking at, hang on, I need to go back to component parts. So we're now going to go back to this and going to look at bit by bit. Right, I'm going to start with, I'm going to do it right to left across the board. So central nervous system is the starting point. And we're going to start with a bit on the brain, hence the brain model and thing on the bench. And then, as I say, we'll come back through the nerves and look at the autonomic nerves at the bottom there, and then come over to the eye. And we'll just see how we sort of get on tomorrow with that. But we're going to do the brain this afternoon. Now, oops, okay. Now, because what we've done in terms of resting potential, action potential, synapses, this is what's going on in the brain. All right, so when you create a memory in your brain, when you learn something and you remember it, you are creating new synapses. All right, that's long-term memory, sort of permanent memories. So, you know, in learning what your name is and how to walk and stuff like that, that's when you're creating new synapses. Now, ideally, you know, when you're training for a profession, you start committing stuff to, you know, actual real permanent memory, don't you? So you don't have to be constantly looking up. So I would say, for me, 99.9% of what I talk about to you is in my permanent memory. So it's all there through synapses. I very rarely have to sort of check out details in notes. But every now and then there's stuff that has sort of got, I don't know, slightly disconnected somehow, or has never actually formed the right number of synapses. And I know where it is in my notes, or I know where it is to look up, and I know which bits to look up, but it just does not come straight back. So, you know, when we're looking at how the brain works, you've got to bear in mind it is all about resting potentials, action potentials, synapses. Yeah, yeah, because it's not, yeah, sort of partially recognising is usually a sort of, yeah, a sort of incompletely formed memory, isn't it? I get it a lot in supermarkets with ex-students. I think I know your face, I have no idea what your name is. But then, you know, you consider several years I'll have perhaps 100 students at a time, various groups and courses and things. I mean, this year I've got, what, 30, 45, 65, 75, about 80 students this year. All right, so yeah, 16 of you. So you're only 20% of what I've got. So yeah, you know, trying to remember everybody. If I encounter any of the BTEC Health and Social Care students outside in Asda or something, there's no way I'm going to get a name pop into my head. And then a few years down the line, I might not even remember you. Wouldn't that be awful? Some people always stick in my mind, so you know, you can always try for that one. But yeah, so you know, you don't always form a complete permanent memory. Now, the starting point here then is, what is a brain? And why hasn't every animal got a brain? And which ones have? Oh, we're not too fussed about that because this is human physiology, but you know, we reckon ourselves to be pretty advanced, don't we? We reckon our brains to be something a bit special compared with other animals. Yeah, not necessarily. It's just that our priorities have been a bit different, haven't they, in terms of sort of, you know, wrecking the earth and fighting with each other. You know, when you look at other animals and their brains are geared up to survival of themselves, and that means ultimately their species, we've gone wrong somewhere. This is the thing, isn't it? You know, we are damaging the planet to a point where we may not be able to survive. We are killing each other off, you know, and other animals don't do this, do they? So, you know, yeah, our brains are great. So, yeah, where have they come from? Well, if you look right sort of back into the sort of simplest organisms, you have nerves, or nerve cells, a few neurons. Then you start to get sensory neurons that can pick up a stimulus like perhaps light or food or water or something like that. And then you might get some of those sensory receptors located at the end of the animal that moves through the environment. So, you know, front end of a worm type thing. So you're starting to get something that isn't a brain, but it's a group of nerve cells. And you've still got a nerve cord, perhaps, that runs the length of the body. So it can communicate, you know, different parts. The more advanced you get with the animals, then the more of these nerve cells you'll have accumulated at the head end. And then you start to develop special sensory organs like simple eyes and things like that. And then once you get into the vertebrate animals, where you've actually got a spine, then we get something that we call a spinal cord. And the top end or front end of that is what we call the brain. And the brain is not the same in all animals. So you have this, at the moment, there's a bit of a debate, isn't there, on sentience in animals. Anybody hear about that in the Brexit negotiations? One of the things, yeah? One of the things that happened, and there was a huge outcry on the internet, was that the UK decided not to go with the European definitions of sentience in animals. Now, sentience is, if you like, awareness. And it has implications in terms of comfort and pain and distress and that sort of thing. So the fact that we were saying, no, we don't agree with the European definitions of an animal's capacity to feel distress and pain and stuff is a definite backward step. And there was a petition and all this sort of thing. But that was people's sort of knee-jerk response. When I looked into that further, it does seem as though the reason we decided not to go with the European definition is we want a better one that's actually better for animal welfare. So, you know, hopefully that means that it will be, you know, we'll consider it in a much better way. But of course, this has implications in terms of transporting animals, keeping animals, slaughtering of animals, you know, how they're kept in zoos, how they're kept as pets, all that sort of stuff. Even down to use of things like perhaps poisons for killing mice and rats, and you know, all that sort of stuff has an impact, doesn't it, then? The only problem is, if we say, no, we don't want the European one, then we are actually without one. You know, we're without a definition, aren't we? So we're in limbo and people could do anything. So I'm not sure what position that's reached, but there are laws in place, but it's, yeah, sort of what it means, I think, is, yeah, it's not clear. That's right, yeah, I mean, shrimps and crabs and lobsters and things can, you know, is it okay to just hurl a live lobster into boiling water? You know, I'm not sure. I really couldn't tell you because I'm not an expert on their nervous systems. But, yeah, you know, I believe so. Yeah, I believe it's more humane, yeah. Yeah, I think, yeah, you should be able to put a knife where the shell is, sort of behind what would be their head, will be where the nerve cord is. So yes, if you cut that nerve cord, you will essentially kill them. Then if you cook them straight away, then yeah, I think no, but that's all part of that, and I think that's not necessarily sort of recognising sentience in a lobster, but I don't know, it's a step in that direction, isn't it? But it depends what areas of the brain an organism's got and how developed they are, because these vary. And you can actually map out which bits of the brain are responsible for what. And, you know, an obvious example would be, well, how big do you think the area is in a human brain to detect smell? Not very big. But how big do you think it is in a dog? Much bigger. So, you know, it's along those lines. You take everything, every aspect that the brain deals with. In different animals, you're going to have different, yeah, amounts of brain. So, yeah, strengths and weaknesses in terms of, you know, its capacity to do stuff. We, however, are just doing the human brain. And if you have a look, there's a single sheet there. One side takes you through a bit about development of the brain, so I'll talk about that in a minute. Down the bottom right of that, there's a diagram that doesn't really belong there, which is about parts of what we call the cerebrum. And then on the other side, we've got a map to fill in, a diagram to colour, if you like. I'll have to do a quick sketch of that to make it work. But on that first side, the development bit of it is interesting. We all start off as a single cell, but that multiplies until we end up as a ball of cells. At that point, all the cells are identical. But somehow you've got to get from a ball of identical cells to something as complex as a human, with skin and muscle and blood and nerves and kidney and all that. So how does it all know to develop in the right place? How come our skeleton's inside? How come it's not around the outside? How come occasionally you get a hiccup where somebody's heart develops outside their body? You know, that's been this week's story. The development's controlled by genes, and sometimes a gene will make a mistake. And these genes are switched on and off at different points in development according to chemicals that are about at the time. So a chemical message or signal at some point takes this ball of cells and determines that in an area on the outside will be the nervous system. And you're just thinking, hang on a minute, but the nervous system's not on the outside, is it? No, but you look at the diagram, you can see what's going to happen. And this is known as the neural plate. So it's an area of cells that are just identified according to their position and chemical gradients and things. They are identified as being the starting point for the nervous system. That then starts to fold in. So the first thing you get is like a sort of dent developing on the surface, like that. And because those cells are there, all right, we start to get this folding in. And then gradually, these little bits will fold in over the top, and you will get a tube running through the length of the embryo called the neural tube, and that's then the basis for the whole nervous system. Oh, it even grew as it went across the board. Wasn't supposed to particularly. But this comes from the outer layer of the embryo. So not surprisingly, so does the skin. So, you know, our nerves and our skin have got origins in common. So that will then form this nerve tube, you know, this nerve cord down the middle of the body, and then the front end will develop into the brain. And if you have a look at the other little set of diagrams there, you've got the little series of 9 images that show you from embryo to fetus to newborn. You can see that what happens at the sort of top end, front end if you like, is that you get an increase in the amount of tissue at the front end, and that then sort of folds over and forms the shape that we're familiar with as a brain. And one of the most significant things you notice is the development of these folds, if you like, and divisions in the surface of the brain. And if a baby's born prematurely, they don't have those. Those develop sort of quite late in pregnancy, and it is just a neural development thing, and I don't know if it's a surface area thing or what, but yeah, it's a sort of fewer folds. Yeah, I don't know. Size-wise, you know, you can only fit in there as much as the skull will allow, really. So yeah. Now, in terms of parts, we need to have a look at that diagram on the next slide and sort out the labels. So just bear with me while I do a quick version of it. Yeah, definitely looks like a peach, that bit. Okay, this should look similar to what you've got, enough that we can actually identify the parts. And I got it all, I think, just about. Oh, I've missed a bit out. Oh no, I haven't, it's there. Right. Now the problem with this diagram is it's two-dimensional. And the brain is very, very three-dimensional. So we have these two models. I mean, this is a, you know, same two-dimensional thing. You know, that's no great help, is it? That thing that's drawn like a peach on there often looks more like a leaf. But of course, the brain is a three-dimensional thing. And what you're looking at is, whoops, okay. What you're looking at is, oh, I can never be a neurosurgeon. Bits of brain are just drop off all over the place. I've lost a bit. Oh, there it is. Right, okay. I can never get it to all fit properly in my hand. Oh, I put the wrong bit in. That's it. All right. you're looking at a three-dimensional thing. So there's an awful lot that goes sort of into the board, if you like, from where we are here. There are three main bits that I want to flag up, and then some other important parts as well. What you've got is a series of labels with their descriptions underneath, but I'm going to highlight three areas in terms of sort of what their main functions are, and yeah, hopefully get everything in. Let's start with the big wrinkly bit. It's called the cerebrum, or the cerebral hemispheres. Hemispheres, they're like half sort of spheres, surprisingly. And you've got labels to these which denote labels to different parts that denote what we call lobes. And these lobes are named according to the bone that lies over the top of them. This one here is the frontal lobe. The one on the roof of the head is the parietal. The one at the back, no, I've not, at least I have left enough space, is the occipital. And then the other bit on here, which is one of the lobes, is this bit here that's hanging down from the side. All right, it looks like it's in the middle of the head, and it's not. It's the temporal lobe from over the other side. Now, each of these can be associated with particular functions, but I'm just going to give you an overview of what the cerebrum does. It's got three main functions overall, one of which is to take sensory input. So there's an area for vision, there's an area for hearing, there's an area for the tip of your right little finger, there's an area for the middle of your left elbow, there's an area for the bottom of your right foot or the back of your left knee. You can actually map it out on there sensory-wise. Similarly, it's in charge of motor control, so it sends out instructions to every bit of the body as well. And you can identify whereabouts there is control for your top lip or your right arm or your left big toe. It's all mapped out on there. So sensory and motor. The other thing that the cerebrum is responsible for is what we lump together as higher mental functions. So this is the bit where we think we're great. But it's things like complex language skills, emotions, behaviour, memories, all that sort of stuff. So that's what the cerebrum does. So I'm going to put that in red down here. Try and keep track of them. So we've got sensory input. We've got motor output. And we've got higher mental functions. Whoops, mental functions. And that is things like memory, emotions, behaviour. Oops. I found some strangely shaped letters today. So that's that bit. And you know, if you look at that diagram on the bottom right corner of the previous page, you'll now see what we've got in there. So you've got in the frontal area, you've got things like thought, reasoning, behaviour and memory. Sense of smell sort of comes in directly behind the nose pretty much. Hearing is right next to your ear. Vision is right at the back of the brain. And then we've got movement and sensation up across the either side of that division at the top there. All right, so you can map it out. Now, how on earth would you find these things out? Well, no, you dissect it. How do you know what it does? Well, what we actually tend to look at is people who've had brain injuries. Yeah? So you have a brain injury and you lose your speech, you look at where the injury is. Oh, that bit does speech. Process of elimination, isn't it? So, you know, a blow to the back of the head can influence your vision. What? Mental. Higher mental functions. What sort of word would marital be? That's why I say it as I do. Whoops. Mental. Yeah, it went a bit lumpy in the middle. So, higher mental functions. Okay, so that's the part of the brain we, you know, are really sort of trying to find out more and more about. It's the, you know, brain in a bottle sort of horror film brain that we're familiar with, isn't it? Now, if we have a look at what else we've got, the bit that's drawn like a peach on there and like a leaf on other diagrams, it's actually a three-dimensional structure like, hang on, I've got to try and hold the two bits together. Like this, it's got, you know, sort of two wings that come out of the side. It's just that the cross section looks a bit like a leaf. Why it's drawn peach shaped on there, I don't know. But that's a bit which is called the cerebellum, and where cerebrum sort of means brain, cerebellum means little brain. And this bit controls voluntary movement. It coordinates voluntary movement. So as we learn to do things like walk, ride a bike, play the piano, you know, anything like that, this is where those programs are stored, and they're the ones that you don't forget. So you don't forget how to walk, you don't forget how to ride a bike. All right, so those programs are stored in here. And some people have phenomenal capacity, don't they, for storing stuff in there. You know, if you're a fan of Strictly Come Dancing, you know, you look at what those professional dancers can store in here. You know, on a weekly basis. Not only are they doing the dances they do with the celebrities, but of course they're doing all the show stuff as well, aren't they? The group dances and stuff.
Yeah, you must have a really big one. So it's not me.
I'm not moving, so it's not me.
No,
No, this is voluntary movement. Movement. This course does not require me to move in any way, actually.
You know, you get those children they call savants, so they can hear a piece of music and they just play.
Sure, yeah. There are, well, connections in the brain are strange things. It's like you also get, yes, apart from being able to learn music by ear, which is that's a sort of thing in itself, you know. Yeah, but they might not be able to remember that and do it again. They might be able to, but it might be that the sequence of movements is remembered somewhere else in their brain and then transferred to here. But yeah, you know, the brain isn't always wired up the same in everybody, I think is, you know, is really important, isn't it? So that bit, where's the label for that? Oh, just up on the right-hand side. So that cerebellum, it coordinates voluntary movement. You'll have a lot of this in the on the sheet anyway. So it's got input from, say, the eyes, the ears, pressure receptors. Then the other bit that I particularly want to flag up is the bit that connects the brain to the spinal cord. As an area, it's known as the brain stem for obvious reasons. You know, you think you've got a nerve cord, a sort of like the stem with a big bit on the top like a flower, so your brain stem is the bit that connects the two. And there are several parts to the brain stem, and there are two parts identified on your diagram. One of which is, let's get these the right way around, the upper part of it is called the pons. Pons means bridge. Right, now if your French is any good or similar, then pont is the French for bridge. So it bridges the brain and spinal cord, and it directs nerve impulses the right way when they come in or go out. It's the bit below that, so it's your bottom label on the left-hand side that's the most important bit, the lowest section of the brainstem at the top of the spinal cord, and it is called Right, we have. I'm going to have to. Right, that bit is the pons. And this bit is the medulla oblongata. Right, medulla.
Yeah.
All right.
Boy.
What?
Water boy.
Water boy? What?
Adam Sandler.
No idea. There's a tendency for people to call it the medulla. It would be if it had got one L in it, wouldn't it? What? How do you pronounce the word that's spelt D-U-L-L? Dull. So it's medulla. Yeah? Medulla. Yeah? So medulla oblongata. When you get a double consonant, it's an English lesson, the double consonant shortens the vowel in front of it. So, for example, over here, this is ok, not oak. O instead of O, and here it's instead of oo. Yeah, I'm a scientist. But these are parts of the brainstem. Now, the pons is, say, a bridge between the brain and the spinal cord. The medulla is the bit that actually keeps us alive, controls our heart and breathing. So that's the bit they look for if they're looking, if somebody's had a severe accident and they're looking for brain death. You can have responses from the cerebrum. People can move their eyes and, appear to be awake, but if they can't support their own breathing and heart, the medulla's gone. That's it.
What about if you get like a blood clot in your brain? Where's that? Is that in that part?
Where?
Or anywhere?
Could be anywhere.
Oh, it could be anywhere.
Could be anywhere.
Couldn't just go to one specific.
Oh, no. Your brain's full of blood, so it could. Yeah, I mean, a blood clot in the brain is a stroke.
Yeah.
Or a bleed is a stroke as well. You've got your different types. And if it cuts off the blood to a particular area, that stops working. So, yeah, if you've got a blood clot to the air...
Like if someone got one in the back bit with movement, then they might not be able to walk properly.
Well, the back bit would be vision, but, yeah, if it went in the back bit, somebody might suddenly go blind. They wouldn't be able to see. So, yeah, you can usually work out where it is by the symptoms somebody's got. But, yeah, if it was in the medulla, it would be... Locked-in syndrome, I don't know, and I don't think anybody knows, and that's why they can't do anything about it. But it's
last week.
yeah,
This week just gone. The guy had a stroke. He ended up with locked-in syndrome.
yeah. It's almost similar to motor neuron disease, isn't it?
Yeah.
Where they've got the incoming information, but they can't do anything, can't respond. It's a scary thing, isn't it? But the brainstem then, really the most important bit and the most primitive bit, really, because that's the bit that without that, nothing else has a function, does it? So brainstem, you've got the pons, and that's our bridge between brain and spinal cord. And then medulla oblongata controls heart and breathing. I'm just walking back and forth here with half a brain in my hand. Now, we've got a few other bits worth flagging up, but those are the sort of main structural parts. So what else have we got left? Well, let's work our way down. There's a curious sort of C-shaped thing in the middle there, with its label on the upper right, second label down on that side. And that is the band of fibres that connects the two sides of the brain. It's called the corpus callosum. And I have read, I think more than once, that this is thicker in women or females, girls, than it is in men. So what that implies is that there's more communication between the two sides of the brain. Are we familiar with that concept? Particularly during communication, if you look at scans in women, communicating different or bits light up on both sides. In men, it's just one bit. And it sort of goes with this idea that women can multitask. There are differences between male and female brains, but it is a spectrum. You haven't just got one or the other. There's all sorts in between. But it sort of goes with this idea of multitasking and the ability of the two sides of the brain to communicate between them. But multitasking... Back to the medulla. Yeah? Controls heart and breathing. Okay, back to... this thing then, the corpus callosum. The idea of women being able to multitask and men can't, it's not quite as simple as that, is it? You know, some of them can. My husband defines my multitasking as the ability to do loads of things at the same time, all of them badly. But, you know, we don't necessarily focus, but we get stuff done. But if you know, you think back to our sort of origins, let's say when we were, I don't know, cavemen, for want of a better word, and men were going out hunting. When we'd reached the hunter-gatherer stage, they call it, men were going out hunting and needing to focus on one particular thing, you know, maybe a wild boar over there they wanted to catch, whereas women would be staying with the children and gathering berries and fruit and stuff like that, and needing to keep an eye on the children, watch out for animals, look for food, keep an eye on this, look at that. So, you know, if you just focused on looking for the berries, your children would run away. So, you know, you've got to multitask as a woman. It helps your survival. Whereas with a man, if he's hunting a wild boar and he's distracted by a few berries, well, they're not going to eat, are they? So, you know, it's very, very basic sort of ideas like that sort of make sense, don't they? That you can, you know, it never is as simple as that. But the corpus callosum links the two sides and allows communication between them. I don't think we've got a lot left on here that's unlabelled, have we? Spinal cord down at the bottom. There. And then what's that thing dangling off there then? It is the pituitary gland. Yep, we've met that before. Can you remember which bit of the brain it's hanging off? It's not labelled on there. I don't know if it's mentioned, is it? No, it's not. If you wanted to put an extra label in, because it will get a mention, do you remember the hypothalamus? That's just the bit above the pituitary, really. And that was where we'd got our connection between the endocrine system and the nervous system. Right, so we've got, I mean, that's a lot more bits than that we could mention. You know, that's a very, very sort of oversimplified view of it. And I'm not going to spend any more time on the brain as a structure. But what you do get in the exam is a diagram of the brain to label bits on and to identify structures related to functions or functions related to structures. But you'll get a similar sort of question in the mock exam, so you'll know what sort of thing you're expecting from that. Now, the brain is only part of the central nervous system. The other part is the spinal cord. And the spinal cord is not actually terribly interesting. It's just a giant bundle of nerves that connects the brain to the rest of the body, brings information in, takes instructions out. And so that's what I'd rather do the brain. The brain's definitely got more going for it than the spinal cord. But what we do need to do is to consider the nature of spinal reflexes. But I think, given the fact that we're several people missing today, I think we will pass on the reflexes for now and pick up on that tomorrow morning, because the